is where we also have the different gatings. You can see the what are the different gatings that we do on a particular sample. And let me just pull out the pointer. Okay, and then also as we go through the gating hierarchy, we'll see the statistics area, the cell number area will change. And what you see here, these are the different keywords. So like HIV status, as I was telling, if you have different patient samples, so this is HIV negative, for example, some of might be positive, then you put those specific criteria that you assign to those patient samples, and they will show up. Like this is a PID, PID is patient ID, this is lab donor 1, all of them are from lab donor 1 here, what you see. And this is a stimulation condition, like NS and PI, what it means, I'll explain in the few coming slides. So this is how you can organize your sample analysis area for better presentation of your data and for your ease of analysis. Okay, so this I was talking about. Somebody's requesting control. I think. Okay.
Yeah, Amlan, please ignore any request for controls.
Okay. Okay. Yeah. Thank you. So if you, for example, here, if you see different keywords that you have created, if you have coined for your particular experiment, so they can be shown here, and you can just double click here and add to the ones that this is the columns to display. If you want some of the keywords, let's say you have 10 keywords, out of which you really want 4 of them to be displayed every time on your workspace, that you have a preference for for doing that under the configuration and under the edit columns. If you double click any one of them, it will be shown. Like these are the just a modification that you can do. And adding a new keyword, how you add different keywords, there is a like this key icon is there. If you click there, there's straight away gives you to the add keyword option, and there you can type in whatever keyword you want, like stim, PID, that as you have seen, HIV status, and all those things. So now this is very interesting, the graph window. Once you start doing analysis on a particular FCS file, let's say here this is a, you can read the, although I know the font is really small, it is LD1 NSNS. This is a particular FCS file which was part of that workspace. Now you double click that, it will open by default FSC versus SSC, and then you can then you can analyze it for the different markers that you wish to. Like here in x-axis we have CD3, and in y-axis we have HLA-DR. And as you can see, the 3 different clusters, this is your CD3 positive T cells, these are HLA-DR positive, most probably these are the antigen presenting cells, monocytes, B cells could be, and these guys are neither of them, so mostly they will be from the natural killer cells. And so this is what we have done, is a gating. And how do you find this option? Here at the top you'll see different gating tools are there. Like this is a rectangular gating tool, this is a quadrant gate, this is an ellipse gate, this is a polygon gate, this is a freehand pencil gate, this is an auto gate, and these are the 2 different variations of a quadrant gate. There's a curly, there's a spider gate. We will use some of them in today's demonstration. Now coming to parameter selection, anytime you want to check different measure parameters on your flow cytometry data, you can just click on this axis here on the X, either on X or Y, and change it to the desired one. So plot view options are there. So this, what you see here, this is a pseudo color. This is called a pseudo color, where you see the most dense antigen dense area is at the center, and as gradually you move to the periphery, you see the color coding changes from red to blue. So, but you have a bunch of different options here, like a dot plot, regular one, then the contour plot, zebra plot, density plot, and even histogram. So now this is what is called active gate. So active gate is now showing events inside. So within the gate, what are the events? You can see this is like a tinted area. You can as well choose the events outside also. Within the, in the FlowJo, the option is there. The events which are outside the gate, that can also be seen if you choose that. So basically this is the area where you do multiple different operations, and you can arrive at those desired display of your visual display of your data. And this is also very important, navigation buttons. Here what you see is, for example, you are at certain stage in a gating hierarchy, and this is your CD3 versus HLA-DR population. Now and you have applied this particular gating tree to the group. So after that, if you want to move to the next sample and see what is the CD3 HLA-DR analysis, you have to click the right arrow. If you want to go to the previous file and see the HLADR versus CD3, click on the left arrow, and this up and down arrow is one level up and one level below. If you want to see the mother population, parent population, and if you want to see the child population from here originating, so you can have to click the up and down arrow for that respectively. So these are the different display options that I was talking about that you have under that graph windows. This particular button, this one, this type of graph that you are uh, viewing. So this is the pseudo color, this is the contour. Basically they are same. You see it is just color coded here in pseudo color. In contour you see the central tendency of that highest antigen density population, and then it as you go to the periphery, it dies down. And these guys, I mean, I can call these guys what you see here as a like outliers. They're not really part of that population per se in terms of the MFI of the antigen expression, but they're just lying on the edges. Yeah, that is what I can interpret. And similarly, density is just a different way of looking to the data. Zebra dot plot is the regular one that I mean we are all used to seeing from the very beginning. And histogram obviously is a univariate plot. Here you just can have one single variable, like here it is CD3. And this is the color map, which is sometimes very useful for the high parameter analysis. Once you have any two populations like CD3 and HLA-DR, you can apply a third parameter on it. It's also called the visual analysis, like a VISNI earlier people used to call it. So you can apply the third parameter and see uh, uh, its expression over all the populations in this 2 dimensions. So gating tools, here you will find the different gating options. I already told there's an ellipse gate, this is a polygon gate, this is an auto gate. Auto gate is very interesting in terms like when you really cannot decide which is the fine boundary of your cells separating from neighboring cells, you click on the auto gate and it will automatically do a magnetic gating around the central tendency of your population. So, I mean, most other options I've already told, and the ones which I have not yet, we'll see during the demonstration. So transforming data. This is a very important feature of FlowJo, and this really makes it a differentiator from the other analysis softwares. And why is that? Sometimes when we import the flow cytometry data and do some few steps into the analysis, we might see in a situation like the data looks like this, what you see on the left side. The main problem with the data is the display. The data, what I see is around the 0 of x and y axis, you see there is a squishing. The data is squished. But I mean, this is okay, fine. The biology will not change. But what you can do, what we can help you do in FlowJo is you can do the transformation so that it becomes visually more appealing. It pleases your eyes, or I mean, whatever, better represent your data and stuff like that. So what we have a transform button which is here for both the axes. Like if this is for the x-axis, this is called T, and here also you find another T. So you click here and then click on the customize axis, and in the customize axis, this window will open where you can really do like little bit of this and that to change the display of that population. For example, you see around 0, it is really squished. That sharp peak you see, you can really extend it, flatten it a little bit so that it appears better, visually better to your and make your analysis beautiful. But having said that, it does not mean that while you transform your data, it changes the data. No, there's no way you can do that. Transforming your data does not change your data. It just makes it look different, prettier. That's it. So these are the different steps that you follow while you're doing some transformation. We will try to do this during our demonstration today. But what you do is once you open the transform window, there are what you see different options like plus minus on the, this is a bi-exponential scale. First of all, let me tell you, this is the scale you see by x. Here you can change it to logarithmic if you want to. So bi-x is what? It's a mix of like linear and log. So around 0, you'll find this, the digits are linear. And if you move further in the positive and negative, you see it becomes logarithmic. So so you see, if you want to have more positive decades, like 10 to the 4, 10 to the 5, 6, you want to have, you can click on the plus. If you want to have less, then minus. Similarly, on this side, plus and minus works the similar way. And then transformation is in the negative decades. If you want to, I mean, stretch your data or pull out this events which are sticking to the axis. you can change this. Even, I mean, this negative will work for you, but even if on top of the negative you're still not satisfied, you want to do some more fine tuning on this area, you can use this extra negative decades. And width basis is something which is like around the zero. If you want to flatten it, if your population is squished, then you can use that, but not necessarily everything you have to use for every data. You have to, I mean, play around with these three options and see in which particular setting your data is looking good, so that you can use it. And before, I mean, there is one good option. If I just go back to my previous slide, suppose this was the point where I first thought that I would do a transformation. And remember here the CD3 is on the x-axis, so you click on the T and click on the customize axis, and then this opens. Now after doing the transformation, you can apply the same transformation to multiple parameters at one go. That's the benefit of it. And then all of them will be having that transformation applied. You don't have to do it for individual parameters. And also, if you want to do the same kind of transformation in your future experiments, provided you have the same marker fluorochrome combinations, and also you do the experiment on the same cytometer, then before applying, you can as well choose to save it here at this option. So saving will also add it under the preferences. You see that $ file name CYT cytometer, and then this will be applied there and it will be saved there. So next time, if you are having the same data set, like you do the experiment twice, thrice, whatever number of times on the same instrument, keeping the marker fluorochrome same, once you import the data and start doing the analysis, it will be, you can apply that same transformation from the preferences. So this is already I told. Effect of transformation, it will not alter your data. The example is this, the three of them are having the same percentage of CD3 cells. So it is 78.4%, but this is the one which I will represent. And this is the one where I see the squishing, I do the transformation, and it could happen the reverse also. You can see some of the events are like sticking to the axis or stuck to the origin. So you want to pull it out, so you do the transformation. Either way, if you have here, if you are here or if you are here, you can arrive here by using the transform button. So I think this is also published in Nature Immunology, this data. A part of the transformation was used by Herzenberg group. So now, this is, I'm quite at the, before entering to the live demo. So I think 2-3 slides I have, which will give you a background of the FCS files that you will be seeing in next half an hour or 45 minutes. So the data set that I'm going to use it is called the Prefix Data. So Prefix is an abbreviation. It is phosphoflow intracellular cytokine staining. So it's a polyclonal prefix assay. So what we did is, I mean, this is done by Tim Crawford and his group and published already in Cytometry. So what they did, they had human PBMCs, they thawed and rested it overnight. And then they did a PMI anamycin, the mitogenic cocktail, they stimulated with for 2 hours first. And at that time, they put some cells which they did not stimulate. This is NS. NS is no stim. So this is your PMI anamycin stimulated, and this is not stimulated for 2 hours. And then what, at the same time, they were doing they were adding some like a monensin or a brefeldin, some of the protein transport inhibitors. Why? Because we want to measure the intracellular cytokines. So they had to be trapped inside the cell and accumulated in a substantial amount so that you can do some assay and see it. That's why you add that protein blocking stuff here at this point. And then what they do is they do the viability staining, and this is a fixable viability dye that we most of us use for experiments where the cells had to be fixed. And then we do the surface antigen staining. So as you can see, there's a T cell panel, CD3, CD4, CD8, all the helper T cells, cytotoxic T cells, and you have the CD38, the HLA-TR, the class 2 HLA. And these are basically the activation, this tell about the activation status of the cell. And then what they do is they again stimulate with PMI anamycin for 20 minutes. It's a short stimulation. And again, at this point, they keep one cell which is not stimulated. So what it actually yields is something what we'll see in the next slide. So just keep in mind, they do two set of stimulation. One is initial for 2 hours and one is later for 20 minutes. And every time they keep one set of cells which is non-stimulated. So after that, I mean, it's the last leg of the experiment is they fix it, they permeabilize and stain for intracellular antigens. This is like a phospho ERK is extracellular signal regulated kinase. So this is, I mean, if this levels are high, it means intracellular signaling is happening inside the cell. which in terms will lead to the gamma interferon production from the cells, and perforin is a cytotoxic marker which is employed by the cells to make holes on this on the target cells which they are going to kill. So these target cells typically would be those like viral infected cells or tumor cells. So that is the basic idea, the experimental plan, and these are the readouts like ERK, alpha gamma, and perforin. And this is in a outline. The experiment is you have PBMC, do the first stimulation, then surface staining and live dead staining, then stimulation 2, then fix it, then do the intracellular staining, and then run on the flow cytometer. So what it essentially means, if you have two different set of stimulations, first is for 2 hours and second is for 20 minutes, the whole experiment boils down to a time course experiment. So you will have 4 different types of cells. One cell which will have no stimulation, either the first stimulation is not there, not the second one. So they have total 0-minute stimulation. So there will be one set of cell which will be 0 minutes stimulated. One set of cells will be having only the second stimulation, the PMI animation. The first stimulation is not present. So second stimulation is for 20 minutes. So they will have total stimulation for 20 minutes. Third set of cells will have first stimulation, not the second one. So it is 120 minutes. And fourth will have both. So it's 140 minutes, 20 plus 120. So basically you can transform the experiment to a time course experiment, 0, 20, 120 and 140. And what you see here is a fantastic kinetics inside the cell which is going on. So the cell which is not stimulated by PMI animation in either time point will show no expression of ERK or the interferon gamma. But the one which has got a second stimulation for 20 minutes, even that brief time point is enough to, give you a whooping amount of this ERK inside the cell, which is fantastic. You see that blue line here. This is the 20-minute stimulation. But IF and gamma response will take, it will require more time. So in the second cell, we don't see any IF and gamma response from the cells. But after that, when you stimulate the cells for 120 minutes and above, it is same. In both the cases, we see both interferon gamma and ERK are expressed and quite at similar levels, which are very high. So this is the stimulation conditions, and you will be seeing in the FCS files these abbreviations, NSNS, NSPI, and all stuff like that. So the last slide in the presentation before I go to the live demo is I think this is last. Yeah, this is the last one. So in total, this experiment is an 8-color experiment. You have 8 different fluorochromes. So and you have different groups. So 20 FCS files will be there which are having all stains, all of them 8. These are the real experimental data files. And 14 are FMO controls, which are fluorescence -1, which we use for gating purpose. So this is a FMO example of a HLA-DR FMO, for example, where you don't add the HLA-DR antibody just to check the absolute background, which is contributed by rest 7 antibodies. Why I call 7? Because if you don't add HLA-DR in an 8-color panel, the rest are 7 antibodies which you add and which gives you an absolute background. From above, for example, above 10 to the 2, I can consider whatever signal comes as HLA-DR positive. So this is all stain, this is HLA-DR FMO, and this is a compensation. So this is not compensated. This is a single CD3 stain file, which is in Alexa 700, and we can see it is like spilling into the APC87 channel. But after you do the compensation of this CD3, it will look similar to this one. Like this is also CD3, it will like a -1 and a +1. So these are also important to generate in a multicolor flow cytometry. We all know that all the detectors that are receiving some signal, they will be receiving signals from other channels also, not from its respective channel only. So you need to do proper compensation, otherwise you'll just you'll get misled and you'll find some MFI which is not proper. So with that, I will end the presentation, and I will now move to the demo part.
Okay. Amran, thank you very much for the presentation. Before you go into the live part, there are some questions which are asked by the participants. Let's address those questions before we get live.
Yes.
So Ankita, could you go through the questions?
Sure. Start.
I see the last one which I can read out. This was a question. Can you explain a little bit more on the FMO? So Amlan, I think Avni wants to know a little bit on the FMO.
Okay. So FMO is fluorescence -1 controls. These are basically gating controls, what we use for a multicolor flow cytometry experiment. So the background is when you have different markers in your assay, some of which the expression pattern like we call a primary antigen, like a CD45, CD348. We know whether it will be either a plus or minus. A positive population will be there, negative will be there. There will be no intermediates. We are very clear about all those markers. So for those, we do not necessarily keep an FMO. It's I mean you can keep it, but it's not required. But the ones which you are not sure about the expression level that you're going to experience in your cells, you better keep an FMO to check the absolute background. Earlier, people used to use the isotype controls, but that idea is little getting backdated now. People are really considering the FMOs a better control for checking the absolute background. This is like if you have, let's say, 4 fluorochromes in a particular experiment, and you really want to check what is the effect of rest 3 fluorochrome on any one fluorochrome. So how do you determine that? You just take a tube and add all those other 3 fluorochromes, but not the ones for which you are making the FMO. So all the other 3 fluorochromes will give you whatever background they want to give, and that you can consider as the absolute background. On over and above that, when you get a signal on the fourth one, the FMO, that marker, that you can consider true positive. I think I have explained
Okay, yeah.
FMOs.
Thanks, Amlan. I think, yeah. So
Yeah.
somebody also ran up with a question saying that is FMO replacing the isotype control? No, isotype control has its own role to play. FMO has its own role to play. FMO is actually going to see the effect of other fluorochrome onto that particular channel. So if I have a 8 color experiment, okay, and I just FMO is full -1, so I have to keep 8 tubes of one less antibody, right? So I'll have a first tube of first like FITC-P per CP. I will have a tube without FITC, but rest all will be there. Then the second would be without P, but rest all will be there. Okay, so we are trying to see that FITC channel, how is it getting influenced by the remaining 7 colors or 6 colors or whatever colors are there in your experiment. Okay, and we want to negate that effect. If your compensation, if your settings are all done well, ideally those fluorochromes should not interfere into this channel, which is negative, and this is what we got to see with that. Okay, so it plays a role, very important role in a multicolor experiment, especially if your color experiments are more than 4 colors. Okay, so mostly in a 2 or 3 color experiment, people generally do not bother so much about the FMOs, but if it's a huge multicolor experiment, definitely we would like to keep a FMO, preferably for all, but as Amlan said, could be those critical ones. Definitely I would like to keep a FMO. Like suppose I want to look at the FOXP3, okay, you do not see a very good separation of FOXP3 from the rest. Okay, so and then whether that little shift, is it FOXP3 or not, it is difficult to see. So if you keep a FMO for FOXP3 in a cytoplasmic staining, it will definitely help you in that experiment. Okay, somebody has also asked question on compensation. I think we are going to talk about it next session, but yes, compensation controls are definitely required, and that's very important when you're doing a multicolor experiment, right? If it is any experiment which is more than single color, 2 colors, you have to have your comp controls so that the machine or the software can do an optimization and the compensation setup correctly, optimally. Ankita, do you see any more questions?
Sir,
Yeah.
I can find one more question which is technical or should be addressed by Dr. Amlan. That
Okay.
is from Shalmali. So she's asking, Amlan sir, can you please explain how to change the width of the histogram? I think she's referring to transform function.
Okay.
And Amlan is anyway going to give a
Yeah.
live demo on that. So I think it will
Yeah.
better
Yeah. I
explain
think
when
that'll
it's
come.
going.
That'll come in the live session, right?
Yes,
Yes.
that would be more appropriate if I show there.
Another thing, Amlan, will you be showing in the live demo that can we go beyond 10.5 frame?
What is it? Sorry.
So,
10
Shikha,
raised to 5. I think she's talking about the fifth log. How do we go
Ah,
about? Yeah.
okay. Yeah, there are options for that. Let me
Yeah.
let me try to find it.
Yeah. Okay. And the next question I just see is importance of live death staining. Could you please explain? Amlan, do you want to take this up?
Yeah, so as you can, it's very evident, like you don't want to gate your dead cells also and trying to find some signal from it. So even, these are like few housekeeping things we do in flow cytometry. We follow these things, like you have to have a doublet discrimination, you have to have a live dead. So live dead dye is very essential to just discriminate your live cells from the dead ones. We Because sometimes I've seen that dead cells do emit signals which are false positive. You If you don't have that live dead discriminator, you might believe that these are real signal, but which is not. So because they have like all those apoptosis and things going on inside, and then they show up certain markers which are like not in the normal expression, and you would might believe that that is a real one. So live dead is very essential. You have to keep a channel separate for that. And if your experiment is very simple, you can have the viability dyes like the 7AAD, which binds to the DNA stoichiometrically. But the ones here we are using are fixable live dead, because if you are anyway going to fix your cells during the course of staining, once a cell is fixed, if you add that viability DNA binding dye, every cell will appear as a dead cell, which is not true. So that's why we use these fixable viability dye, which are basically amine reactive dye. They react with the amine group NH2, and they form those. They bind to those groups, and they give you emit a bright fluorescence. So the ones which is a dead cell will have a compromised plasma membrane, like a leaky one. So this will go inside and bind to the amine group and give a brighter fluorescence. So the idea is the ones which are like live dead negative, or here is like this is the one where they used AARD dye. So AARD negative, you'll see that we will take that as live cells.
Amlan,